Okay, so we are going to look at the neuron. So we're in chapter 10, the general one of the nervous system. We're looking at the neuron. This is slides 7 through 15. So this picture should look familiar. This should be similar to the one you see in lab or whatever. This is the one in the textbook. This is a picture of a neuron, a general idea of a neuron. So like we said, the neurons have a couple of special things. First of all, we have this cell body. We have an area of neurofilaments, and we're going to find out a little more about them. These are structures. They are going to be found in an area called the axonal hillock. All the axonal hillock is where the axon is leaving the cell body. All right, so this general area right here is the cell body, and as the axon is leaving, this has a special name, like we said, the axonal hillock. And inside of it, there are these little protein fibers called neurofibrils, and they are there to help guide things, guide the impulses to this little area. So we talked about dendrites, how these dendrites are taking information into the cell body from all over, and all of it's being funneled down to this area, like we said, called the axonal hillock. The axonal hillock's like a speed bump, basically. It's the starting gate. So when all of these impulses that are coming in from the dendrites reach a certain intensity at that level, then there's going to be a signal that's sent out on the axon. So on this picture, just wanting to point out these two structures here, axonal hillock and the neurofibrils. Now this thing inside the cell body, this chromatophilic substance that we call Nissl bodies, these are these little green spots inside the cell body in here. What I will tell you is I am not going to ask you any questions on those. I will make sure that the test that I put together doesn't have that in there, and if it does, I will take that out. We don't know exactly what it is. There are some ideas, but for us right now, leaving that out. So I want you to know the cell body in this area called the axonal hillock has neurofibrils in it. So we just talked about the axonal hillock, and the axon, like we said, is only going to have one branch that comes off the cell body, but it can branch out, and those are called collaterals. And where they end is a thing called a synaptic knob, and also the axonal terminal. So as the axon comes out, it's going to form this little bulb, and that little bulb is going to release things we're going to look at a little later on called neurotransmitters, little chemicals that are going to go to other cells and tell those other cells, what to do if that needs to produce something or, whatever. It's communication, just like we said, that's what neurons are for. The other thing is that these neurons, a lot of them, their axon will have something called myelin on it. And you might have heard of this before. Myelin, and what is called a myelin sheath, are these things in this picture. They are called Schwann cells. Here's a little name for it. But the myelin is this little area of the Schwann cell that is layer upon layer of the cell membrane of that Schwann cell. So these Schwann cells are going to be, we're going to find out and learn that they're in the peripheral nervous system. They are helper cells. That's why they're called neuroglial cells. And so these Schwann cells are going to wrap around the axon, and they're going to perform a little like insulation around it, and they're going to help send this signal that's on the axon much faster, because where these Schwann cells are, as it says, they wrap tightly around the axon, and they are composed of layers of myelin. All right, we don't need to know it's a lipoprotein, but hopefully then you see this, that you can kind of tell that it is a lipid and a protein mixed together, and it's going to have very unique properties. So this is called the myelin sheath, as it says here, and in between where those Schwann cells are little openings, little areas in between the Schwann cells where the axon is exposed, and those are called the nodes of Ranvier. The nodes of Ranvier. And what happens is, as this impulse goes, it literally jumps instantaneously to this node, and then instantaneously jumps to the next node, and so forth and so on. And so if I had an axon and it was wrapped up with all of these little Schwann cells, instead of the impulse having to travel the full length of the axon, it would be like these little areas are erased, and I end up just having to travel the equivalent of adding up all the nodes of Ranvier, and the nodes of Ranvier are much shorter than that. So it would be the equivalent of traveling this far instead of this far in the length. So hopefully that makes sense. But we'll talk about this more as we go on. Here on this little picture in the middle, you can see this layer upon layer of the cell membrane that's wrapped up over and over again, kind of like if you rolled up a toothpaste tube, an old school toothpaste tube. So we'd have this layer that is called myelin, or the myelin sheath, that is just a bunch of different layers of the cell membrane of this thing that we call the Schwann cell. And this outer bubble of cytoplasm, right, that's right here, that's all around the outside, is called a neurolemma. And you'll need to know that. It's not on here, but you'll need to know that, and you'll know it for lab. This Schwann cell is going to have these two parts. And if it was something else, we'd call it, a lot of other things would end up calling it the outer layer of the cortex, the inner layer of the medulla. And here, the inner layer is the myelin sheath, and this outer layer is called the neurolemma. Now, not all axons are myelinated. We will have both myelinated and unmyelinated axons. In the peripheral nervous system, the Schwann cells will line up along the axon, like that picture we just looked at. Sometimes we'll have Schwann cells, but they'll only be encased in the sarcoplasm. They won't have any of the myelin around it, so it's considered an unmyelinated axon. Now again, we're not going to worry too much about these. What I want you to know, and what's the hardest thing to get to really figure out, is how the myelinated axons with the Schwann cells and the myelin work. So now we're going to look at classification of neurons, and this is specifically a classification by structure. So the structure is going to be how many things come off of the cell body. All right, so I'm going to kind of try to circle the cell body as best I can here. On this first picture, I've got multiple dendrites coming into the cell body and one axon going away. So I have many things that are attached to the cell body. So it is called a multipolar neuron. And as you see here, the majority of all neurons are multipolar neurons. Now I'm going to talk about each of them in general as we go on, but bipolar simply means I've got two things attached to the cell body. So I've got a dendrite coming in, that's number one, and then I've got an axon going away, that's number two. So this is called a bipolar neuron, because it's got two things attached to the cell body. And then we have these kind of different ones where I only have this one attachment here. It is pretty much entirely, as it shows here, the axon, and that the dendrite is simply the end part to it. This, because there's only one thing here, this is called a unipolar neuron. All right, so now some things about these. So the multipolar neuron, as I said here, this is the majority of all neurons, and it is pretty much all the neurons of the central nervous system. Not all of them, but pretty close. All right, so this one right here, and this is the one we use to look at in lab, and in our model is of this. This is a multipolar neuron. It is the most common, and it is pretty much the one, the neuron of the central nervous system. and also it is the motor neuron. And as I said before, this is a tough thing to use to spell here. But motor neurons, that means that the ones sending signals out for your muscles to contract, are going to be multipolar neurons. Now we're going to look at, and I'll erase some of this, we're going to look at the bipolar neurons. Now the bipolar neurons are very interesting. This has nothing to do with the disorder bipolar, but this, again, simply describing the things attached to the cell body. But these are very special. They are the least common, and because of that, that means they're special, but they are these special senses. This is your sight. So when we look at your rods and cones, the photoreceptors inside the eye, they're going to be bipolar. The senses that allow you to smell, your olfactory nerves, they are bipolar neurons. So the bipolar are very special. They are your special senses. And then we get to this unipolar. These are your regularly sensory neurons. There's only one process, like we said, the process right here, and it is mainly all axon. It is that the dendrite is pretty much this little end right here. So imagine that this is in your big toe, and you hit your big toe on something. These little guys here that are called the dendrites are going to feel it. They're going to send a signal all the way up into your spinal cord. So this section right here could be as long as your entire leg, all the way up through the lower part of your low back. When this sensory neuron enters into the vertebral canal, you'll see when we look at the cross-section of the spinal cord, we're going to learn a structure called the dorsal root ganglion. That's where all of these cell bodies are going to form, so stay and hang out. And then this goes into the central nervous system and drops off its message, and it's going to say, ouch, I stubbed my toe. All right, so multipolar, bipolar, unipolar. Multipolar have multiple things on the cell body. Bipolar only has two. Unipolar only has one. Multipolar is the most common. It is the neuron of the central nervous system. It is motor neurons. Bipolar is special senses, your sight, your hearing, your taste, your smell. And then unipolar is going to be your normal sensory. This is going to be bringing in, if you touch something with your fingers, it's going to be going back into your central nervous system by way of a unipolar neuron. And remember, this is classification by strength. Now we look at classification by function. Classification by function is basically saying what it's going to do. So we have, again, three parts, and we kind of looked at this in the earlier one because it's the pattern that things are going to run on. I have sensory neurons, right? These are neurons that are bringing impulses into the central nervous system. Peripheral nervous system, they're sending sensory information into the central nervous system. Now, in general, this is going to be a unipolar neuron, like we just saw. Not all of them, but in general. As it says here, most are unipolar. The special senses that we're going to talk about that are sensory are bipolar. These send their senses, this information, into the central nervous system, and here we see something that is really amazing. These things are called interneurons. Interneurons are part of the central nervous system, right? They are going to be mainly multipolar, and their job is to take this incoming information and analyze it. They're going to go through your memory bank. They're going to go through your emotional bank, and it's going to say, has this happened before? What did we do in response to it? Did we like it? Did we not like it? Do we want to try something different, or we want to do the same thing? So these interneurons are really special, but if something happens and it needs to have something done to counterbalance it, the interneuron sends a signal to a motor neuron. Now, Later on, we're going to look at a reflex arc, and this is the basic makeup of a reflex arc. This motor neuron, which is going to be a multipolar neuron that's going away from the central nervous system. It's multipolar because the cell body is going to be in the central nervous system. And it's going to go to things called effectors. We'll look at this a little later on. An effector is either a muscle or a gland. So a motor neuron is going to make something happen. I'm either going to cause movement through a muscle, or I'm going to cause the release of a substance through a gland. All right. So let's say the air conditioner clicks on in lab and you get cold. Sensory neurons are going to go into the central nervous system, where interneurons are going to get that signal. They're going to say, hey, this happens all the time, so let's put a coat on. That's going to send a signal out on a motor neuron for you to put a coat on. Unconsciously, in the autonomic nervous system, the motor neuron, if you get cold, is going to go to those little erector pili muscles that are attached to the hairs in your integumentary system and give you goosebumps, trying to warm you up. So that's an idea of the pathway. This is the classification, again, by function. Now, neurons aren't the only nerves in the nervous system. We're going to have special helper cells, and these are called neuroglial cells. Neuroglial cells will have structural support, but they'll also help give nourishment to neurons. They will make sure that the ion levels are in proper order. If there's too much, they'll get rid of them. They will deal with neurotransmitters as the neuron sends signals from one neuron to the next. And so they help, and they're mainly going to be found in the areas, what we call synapses, where the one nerve comes in, you get the little synaptic bulb, and it's going to release the neurotransmitters for the other cell to pick up and do something with. All right, we looked at the neuromuscular junction in the last chapter, looking at the way muscles are in, the way muscles contract. So in the neuroglial cells, there are two types. There are neuroglial cells in the CNS, central nervous system, and then the peripheral nervous system. There are four that we look at in the central nervous system. The first one is called astrocyte. Now remember, cyte means cell. Astro should give you the idea of a star, because that's what it looks like. It is a star-shaped cell. They are going to help connect neurons to blood vessels, and that should make you understand. I mean, you should automatically see if it's connecting one cell to a blood vessel, it is going to be for nutrients, right? It's going to aid in metabolism, the breakdown or buildup of certain substances, whether it's a neurotransmitter or whatever. It's going to help regulate ion concentration, such as potassium. Potassium's a big thing. And by doing that and regulating these, it makes part of the blood-brain barrier, which again kind of goes to, it's going to be the connection between the neurons and the blood vessels. In the brain, in the cranial cavity, there is a tighter filter, and it's these cells that provide it. So a couple things. Remember, they are star-shaped cells, and if you wanted, one of the ways I remembered this was they're kind of like the maid, all right? They are the maid or the butler for the cell. They're the ones that are there, and I'm not going to be able to spell that all out, but the maid or the butler, they're the ones that are going to be taking care of these cells and making sure that their bed is made and their food's right. I'm going to jump down to the microglial cell. I'm going to come back to oligodendrocytes in a minute, because the microglial cell is kind of associated a little bit with the astrocytes. They are kind of like the janitor of the neuroglial cells. They're going to help the astrocytes, but they're going to do it by engulfing and getting rid of all of the byproducts, all of the waste substances. So the astrocytes and the microglial cells work together to help make sure the environment of the neuron is in its best possible order. So I always like to remember those two together. So the astrocytes, I always remember it again as like the butler or the maid, the microglial cells as the janitor, because that's a big deal that they're phagocytic. Now, the ones on the bottom, the epidemia or the epidemial cells, these are always associated with cerebral spinal fluid, CSF, right? I'm going to imagine that most everybody has heard of CSF, cerebral spinal fluid. Whenever you hear epidemial cells, you should think of cerebral spinal fluid, because what these do, they cover the ventricles of the brain, which is where CSF is found, and they have cilia on top of it. So I've got this cell and I've got all this cilia on top of it, and what it does is this cilia is going to wave back and forth, and it's going to cause a current. It's going to cause the cerebral spinal fluid to flow. And so if this is the brain and spinal cord right here, it's going to cause this cerebral spinal fluid to flow up and down and throughout the brain and then back around. And so it's kind of, these are kind of the boat motor for the cerebral spinal fluid, getting it to move around to where it needs to go. And so the last one is back up here where we get into this thing called an oligodendrocyte. The oligodendrocyte is the central nervous system's equivalent to the Schwann cell. All right? The Schwann cell, which we looked at, provided myelin to the axons. They are not found in the central nervous system. It is the oligodendrocytes that are the ones that are found in the central nervous system. And the difference is with these, they're like octopus, or octopi, I guess. So if I imagine that these little lines right here are axons, right? So if I drew a little crazy neuron here, trying to draw. I am sorry that these things are so bad, but anyway, I'm going to draw this little cell right here, and this is an oligodendrocyte. And the way it works is it's going to reach arms around and wrap and myelinate different axons at different areas. And so it really is like I said, like a little octopus. And this is a big difference because like let's say we're looking at this one is the peripheral nervous system, PNS. So in the peripheral nervous system, I have an individual cell here, an individual cell here, an individual cell here, and so forth, right? So everywhere that a Schwann cell is an individual cell. On these in the central nervous system, the oligodendrocytes provide myelination to multiple axons for multiple different spots. And this is going to be a big thing when we look at the regeneration of axons if they are damaged, all right? Because these being individual cells are going to act different. So like just for instance, as I get in, we're going to look at this. Let's say that this axon, well, since this is the cell bottle, we'll say the axon is damaged right here. Well, the axon's going to waste away and it'll just disappear. And so I'll have a little bit of the axon. I shouldn't have, say the axon's right there. But the axon that was in this area is gone. And because of that, this oligodendrocyte here is just going to pull this arm back and it will basically erase this area. So when this axon, the one that got damaged here, tries to replace itself, there's nowhere for it to go because it doesn't have a framework for it. So a damage to an axon in the central nervous system it'll never heal because it loses it. Now, the difference is on the peripheral nervous system, because each one of these down here are going to be individual cells. If I damage the axon, and let's say the axon disappears, those cells will hang out for a little bit. And so when the neuron starts to regenerate these, it's going to have a little tunnel that it already knows where to go. So in the peripheral nervous system, the axons can heal. Now we'll talk a little more about that later on, but I hope that makes sense. But oligodendrocytes are the central nervous system counterpart to the Schwann cell. This picture, this kind of shows an or an astrocyte here. It kind of looks like a little star shape. It's going to have suckers out there that are trying to connect. See, this is a capillary, and then this is the neuron, so it's trying to form connections to the two. These are the oligodendrocytes. Again, can have multiple, and when you say multiple, I mean multiple little appendages that go out to provide myelination for the different axons. These are microglial cells, and then these were the epidemial cells with their little cilia. This would be a cavity filled with cerebral spinal fluid. Now, in the neuroglial cells of the peripheral nervous system, there's only two. The Schwann cell, again, is going to be the one that provides myelination for the axons there. Don't forget, we looked at just a minute ago that the gaps between the Schwann cells where the axon is exposed is called the node of Ranvier. But they greatly speed up the speed of nerve impulses, and we'll kind of discuss how that happens. But the other one is called a satellite cell. And the satellite cell does the job of both the microglial cell and the astrocyte. They're going to be always located around the cell bodies, which is where this cell body here is going to have a lot of other axons coming into it. And so and the dendrites bringing information into it. So these satellite cells are going to be hanging out in this area right around in here, trying to make sure everything is in its proper order. All of the chemical concentrations and whatever are working right.